Hello pupils, this is Dr. Lyons again, and what we're going to be getting into in this video lecture is the second part of the genetics chapter. So in the first part of the genetics chapter, we learned a little bit about Gregor Mendel, we learned about some of the experiments that he did on pea plants, and we got kind of an introduction into genetics and into heredity. In the second part of chapter 9, we're going to go into a little bit more complexity with the ways that genetics can work. And so, as with the first part of chapter 9, in the introductory photo, I don't have a picture of genes, but I do have a picture of this cute little thing, which is known as a nudibranch. So this is a small thing that's kind of related to sea slugs and crawls around on the sea floor around coral reefs. So I want to start this video lecture by having you think a little bit about some applications of genetics, right? So thinking about some ways that we could potentially use genetics to our advantage. And so one way that we can kind of use genetics to our advantage and can apply it is with family pedigrees. So a family pedigree is used to do some important things. One thing that it does is it looks at reproduction that occurred in the past. So it basically looks at, who did the deed with who were the offspring that came about from those people doing the deed, and what are the traits that those people have, parents and offspring and grandparents and great grandkids and all of that. So it's kind of looking at the people that are in a family and the traits that they have and how those people are related to each other. With that information, one can often make predictions about what might happen in the future. So if you look at the family pedigree of two different people that are considering having children, you can sometimes make some predictions about what their offspring might look like and what sort of traits those offspring might have. So this is a family pedigree, and a little bit about how things are represented here. So all of the squares that you see are males, and all of the circles are females. And in this case, we're looking at earlobes and whether they're attached or detached. So if your earlobe attaches all the way at the bottom to your head, that is an attached earlobe, and that is a recessive trait. Whereas if the bottom of your earlobe is dangling, then this is the free form where the earlobe's not attached, and that is considered dominant. So it's kind of hard to tell this just by wiggling your ear. You're just gonna have to go to a mirror in your house and take a look at your ear to decide whether or not you have an attached or a free one. And so what's going on here is it's showing us, who did the dirty with who. So Aaron and Betty did the dirty with each other, and they had 4 kids: Evelyn, Fred, Gabe, and Hal. And we can see what phenotypes those kids had, right? So Evelyn has a free earlobe, Hal has a free earlobe, Fred and Gabe have attached earlobes. And then we see Cletus and Debbie, they got together and they made Ina and Julie. And then Ina, the kid of Cletus and Debbie, had kids with Hal. Hal is the kid of Aaron and Betty. And they produced Kevin and Lisa. Okay. So that's kind of what's being showed on this family tree. So we can kind of look at a family tree, and sometimes we can see when something might be suspicious, right? So for instance, here we have a hypothetical family tree with Bill and Betty, who have kids Stacy, Fred, Gabe, and Hal. So Bill and Betty both have the recessive traits, okay? And because they both have recessive traits, that means that they both have 2 copies of the recessive allele. So remember back to the first part of chapter 9 in the chapter 9a video lecture, we talked about alleles and how there are recessive alleles and dominant alleles. And in order to show that recessive allele, you have to have two copies of it. So in order to have attached earlobes, you have to have two copies of the recessive allele. Whereas if you have free hanging earlobes, that means you could either have two copies of the dominant allele, or you could be heterozygous, meaning you have one copy of the dominant and one copy of the recessive. So think about it. Bill and Betty, they have two copies of the recessive allele. So what looks suspicious here? Right, so you should probably all be looking at Hal, and you see that Hal has the dominant phenotype. So he has a free hanging earlobe, meaning that Hal has at least one copy of the dominant allele. But Bill and Betty, they each have two copies of the recessive allele, meaning that Hal must have gotten that dominant allele from somewhere else. So you should all at this point be saying, ooh, what happened here? And so it's most likely that either Hal is adopted or perhaps there was some infidelity going on here. Either way, Hal cannot be the kid of Bill and Betty. So before you all go and look at your parents and look at your own earlobes, just a quick caveat, earlobes are not completely dominant and recessive, right? This is kind of a hypothetical situation, right? So I don't want you going to your parents and looking at their earlobes and then saying, aha, mom, you cheated on dad. I don't want you to do that because it's not quite like that. Okay, so that's how a family pedigree works. And of course, with earlobes, it's like who gives a crap about that? But with other things like cystic fibrosis or sickle cell anemia, with those sorts of things, you can look at a family pedigree and then tell whether two people have a chance of having a kid that has cystic fibrosis or sickle cell anemia. So with earlobes, it doesn't really matter. It's really just cosmetic. But in terms of very serious genetic conditions like cystic fibrosis and sickle cell anemia, family pedigrees can be really, really useful. So let's talk a little bit about genetic disorders such as cystic fibrosis or sickle cell anemia or albinism or even certain types of Alzheimer's disease. So there are certain genetic disorders that behave in the ways that we talked about with pea plants and Gregor Mendel and all of that stuff. So these are genetic disorders that are controlled by a single gene, and it's inherited in a dominant or recessive sort of pattern, right? So it's just one gene, and then there's just two alleles, a dominant allele and a recessive allele. In most of these cases, these genetic disorders are said to be recessive. What that means is that in order to have that genetic disorder, you have to have two copies of the recessive allele. So for instance, to have sickle cell anemia, you have to have two copies of the recessive allele. In order to have cystic fibrosis, again, you have to have two copies of the recessive allele. So like I was just saying, two copies of the recessive allele gives you the disorder. So that then brings up a term that probably a lot of you have heard before, and the term is carrier, right? So what is a carrier in the context of a genetic disorder such as cystic fibrosis? Well, what a carrier describes is a heterozygous individual. So a heterozygous individual has a copy of the dominant allele and a copy of the recessive allele. So, but because they have that one dominant copy, that means that they don't have the genetic disorder. So for instance, a person can have one copy of the recessive allele for cystic fibrosis, but not actually have the genetic disorder, not have cystic fibrosis. They carry the recessive allele, but they don't have it because they also have the dominant allele, which keeps them from having that genetic disorder. So carriers are individuals that are heterozygous, essentially. So why don't we do an example and kind of work through an example with this. So the example that we're going to do is in the case of sickle cell anemia. So I just taught you about how sickle cell anemia is a recessive disorder. So in order to have sickle cell anemia, you need to have two copies of the recessive allele. So let's say we have two parents and each of those parents are carriers, meaning that neither of the parents have sickle cell anemia, but they each carry one copy of the recessive allele. Right, so we have parent 1 on the top, and we're talking about we're talking about blood cells here, right? So maybe we'll use the letter capital B is going to be blood cells that are round, have that nice round shape. And lowercase b is going to be recessive, and that's going to be blood cells that have the sort of sickle shape. Okay. So parent number 1 is heterozygous, meaning that they have a capital B and they have a lowercase b. Parent number 2, same thing, right? So they have a capital B and a lowercase b. Right, so you can probably already tell how I set this up, that we're going to do a Punnett square. So I'm going to draw in my Punnett square with 4 boxes in the middle. Okay, and then just like I taught you in the last lecture video, we're going to fill in those boxes going down and going across. Right, so we fill in capital B, we fill in lowercase b, and then we go across, filling in capital B and capital B, and then lowercase b and lowercase b. Okay, so we can see that we end up with one individual or one potential offspring that would be capital B, right? So because they have 2 copies of the dominant allele, that means that they're going to have round blood cells. There are 2 boxes that have capital B, lowercase b, so those are heterozygous individuals. So again, they are going to have round blood cells. But then we have one box that is lowercase b, and so that is going to then be an offspring that has sickle-celled red blood cells. So we see just one box out of the 4 boxes results in a child that has sickle cell anemia, meaning that that for these 2 parents there is a 1 in 4 chance of having an offspring that has sickle cell anemia. Or put another way, a 25% chance. Okay. So that's kind of a scary thing, right? With these genetic disorders, you can have 2 people that don't have the condition at all, but they can actually have a kid that has the genetic disorder, that has sickle cell anemia or has cystic fibrosis. Of course, the chances are still pretty low, just because there's not even a lot of carriers that are in the population for cystic fibrosis or for sickle cell anemia. But they are there, and occasionally those people come together and have kids, and occasionally that 1 in 4 chance happens where they have a kid that has a sickle cell anemia or cystic fibrosis. Okay, so what I want to talk about now is some variations on Mendel's law, because a lot of the things that we have talked about up until now, has been kind of the simplest varieties of genetics, because what Mendel described with pea plants doesn't actually apply to every single living thing and every single trait of every living thing. It certainly does apply to things like flower color in pea plants or to cystic fibrosis. But in fact, genetics is a lot more complicated than that. There's a lot of variations on what Mendel found that exist in nature. So we're going to talk about that here. Right, so Mendel's laws, those 4 laws that we talked about, they don't explain all the patterns of genetic inheritance that we have. And this will actually be a little bit intuitive when you think about certain, traits. So think about human height, for instance, right? So if you think about, the population of humans out there, there was a variation of heights. So we span all the way from very, very short to very, very tall in all the heights that are in between. However, if human height was determined by Mendel's laws, where there is, say, a tall allele and a short allele, humans would all be either tall or short. There wouldn't be anything in between. But we know that that is obviously not the case. There is people that are of medium height, So that shows us right off the bat that Mendel's laws don't really account for everything. And so human height is one example of that, something that isn't accounted for by Mendel's laws. So in fact, a lot of, with most living things, characteristics that are considered Mendelian, things that follow Mendel's four laws, are actually quite rare. Most of the traits that living things have actually follow a slightly more complicated pattern. So in humans, for instance, there are things like cystic fibrosis and sickle cell anemia. There are certain like taste buds that we have that follow this pattern. But in fact, most of the things that make us who we are just are a lot more complicated than the laws that Mendel came up with. Which is not to say that Mendel's a dum-dum, and not to say that his work is useless, because his work is quite foundational for the field of genetics, but things are just a little bit more complicated than that. If Mendel had studied some other organisms aside from pea plants, he would have found that things are a lot more complicated than what he found with the pea plants. So one way that genetics can vary from what Mendel found is with something known as incomplete dominance. So that is when individuals that are heterozygous, they have a blended appearance between the two different alleles. So up until now, we've been talking about a dominant allele and a recessive allele. But sometimes that doesn't exist. Sometimes just two alleles are kind of are incompletely dominant to each other. It's not as if there's one that dominates the other one. And that is the case with a lot of flowers, for instance. So with a lot of flowers, if you take a red one and a white one and breed them together, you'll end up with offspring that are pink flowers, where these heterozygous individuals, they kind of express both of the traits. They express both the red allele and the white allele. And when you blend them together, you end up with pink. So that's what is known as incomplete dominance, because there's not a dominant allele and a recessive allele. Then there can be, there's some circumstances. In one example of this is blood types, which I'll get to in a minute. There are some certain circumstances where there's more than two alleles. So up until now, we've been talking about there just being two alleles. So say like a tall allele and a short allele, in no other alleles. So there are, in fact, for many types of traits, a lot of different alleles in that population of living things that controls the characteristics of those living things. And sometimes those alleles can be co-dominant to each other. So let me explain that. So this is what goes on with blood groups of humans. So in humans, there are three different alleles that control a single gene, meaning what blood type you have. And we're talking about just the letter part, not the positive negative thing. I'll get to that in a little bit. I think most of you have heard that your blood type is like maybe like A positive or O positive or B negative. We're talking about just the A or the B or the O here. Well, I'll tell you about the positive and negative thing in a minute. So among humans, there are four different phenotypes, right? So you can have either type A blood, you can have type B blood, you can have AB blood, or you can have O blood. In those four phenotypes result from the combinations of three different alleles. And so what these letters actually refer to are carbohydrates. So you'll remember way back from chapter 3 that carbohydrates are sugars. And there are certain sugars that are found on the surface of our cells, including our red blood cells. And so there are two different carbohydrates. There's the A carbohydrate and the B carbohydrate. And so if you have type A blood, you have the A carbohydrate on the surface of your red blood cells. If you have B blood, you have the B carbohydrate on the surface of your red blood cells. If you are AB, that means you have both carbohydrates on the surface of your blood. And you can probably guess what comes next. If you're type O blood, that means you have neither type of carbohydrate on the surface of your blood cells. So where that becomes really, really important is how our bodies react to blood. So if you are type A blood, your body is used to having red blood cells with the A carbohydrate on the surface of it, right? Your body sees those A carbohydrates and it says, oh, that's fine, I'm used to that. That is just normal to me. But let's say, for instance, you are type A blood and you, I don't know, God forbid, got in a car accident and needed a blood transfusion. If the doctors screwed up and put type B blood inside of you, that would be really, really bad. And why it would be so bad is because your body would be used to only the A carbohydrate. If all of a sudden there is B blood inside of you, your body is going to see those B carbohydrates and say, what the hell is that? That is not something good. I'm going to attack that and try to kill it and get rid of it. So basically your body goes on sort of immune system overdrive where it's just trying to kill all of those blood cells that are in your circulatory system, right? So that's really bad. It can be fatal. It can kill you. So that's why doctors have to be really careful with blood transfusions because if you put the wrong type of blood in the wrong person, then it can actually kill them. I had a student that this happened to him when he was serving in the military. Doctors put the wrong blood type in him and it nearly killed him. So now he has tattooed on his hand his blood type just to make sure that the doctors don't screw up again. So how this works is that the A allele and the B allele are co-dominant and the O allele is recessive. And so I'm going to explain how that works with a chart on the next slide. And I already talked about blood transfusions and why that's so important. Okay, so let's take a look at this chart. So what you see here are the different phenotypes, right? So type A blood, B blood, AB blood, or O blood. And here are the genotypes that are possible. So as I was saying before, there are three different types of alleles that a person can have. So there are three alleles that exist in the human population. There is capital I A, there is capital I B, and there is lowercase I. Right, so we can combine those three different alleles in 6 different ways. Right, we can match two capital IAs with each other. We can match a capital IA with a lowercase I. We can match two capital IBs with each other, or a capital IB with a lowercase I. We can match a capital IA with a capital IB, or finally two lowercase i's can be matched with each other. And so the capital I should indicate to you that that's dominant, whereas the lowercase I, that is recessive. The capital IB also dominant, lowercase I still recessive. So if you have two capital IA alleles, that means you have the A carbohydrate. Or if you have a capital IA with a lowercase I, you still have the A carbohydrate on your blood cells because capital IA dominates lowercase I. We see the same pattern with B. If you have two capital IBs, then you have the B carbohydrate. Or if you have capital IB matched with the lowercase I, you still have the B carbohydrate. Now this is where we see the codominance of the alleles, right? So capital IA and capital IB are not dominant to each other, right? They are codominant. So if you have that genotype, then that means you have both the A carbohydrate and the B carbohydrate on the surface of your blood. And if you have two copies of the lowercase I, that means that you don't have a genotype, you don't have genotypes that give you any carbohydrate. So the blood cells would just be completely devoid of carbohydrates. Okay, so let's think about blood transfusions a little bit more and think about, who would react badly to which blood and who can donate blood to who else. So let's say that you are type A blood, so your blood cells have the A carbohydrate on them. Think about that for a minute. Which of the other blood types would you react badly to? So you would react badly to B, and you would react badly to AB. So the B one, that makes sense. AB might be a little more confusing to you, but think about it. So if your body has these blood cells, the carbohydrate A blood cells, your body is only used to these A carbohydrates. If you get a transfusion of AB blood, your body sees the A carbohydrate and says, oh, that's fine, that's what I'm used to. But it also sees the B carbohydrate and it says, what the hell is that? I don't like that. I'm going to fight that right now. So AB blood cannot be transfused into a person that has A blood. Okay, now let's think about the B carbohydrate people, right? So if you have type B blood, you're going to react badly to A, as is pretty intuitive, and also to AB, because as I was explaining before, if you have B carbohydrate blood, your body is used to the B carbohydrate, but not used to the A carbohydrate. So even though the type AB blood, does have the B carbohydrate on it, it also has the A carbohydrate on it. So if you have type B blood, you would react badly to AB blood because your body would see the A carbohydrate and freak out and say, I don't like that. Okay, let's think about a person that has AB blood. Who would they react badly to? All right, so think about this. If you have AB blood, your body is used to the A carbohydrate and it's used to the B carbohydrate. So essentially, you're used to everything, meaning that if you are AB blood, your body doesn't react badly to any blood types, which is great for you. That means that you can get a transfusion from anybody else, right? Which is really, really useful. And that's opposite to these people, the type O people, right? So if you're type O, think about it, your body's not used to the A carbohydrate, it's not used to the B carbohydrate either. So your body can't get blood from an A person, can't get blood from a B person, can't get blood from an AB person. Your body reacts badly to everybody except for O. So if you're type O blood, sucks for you because your body reacts badly to everybody else aside from people that have O blood. Okay, so now let's think about who can donate to who. So if you have type A blood, who can you donate that blood to? Well, you can donate to other people that have A blood, and you can donate to people that have AB blood. Because the AB people, they don't react badly to any blood. They can take it all. They're just fine taking any other type of blood. And if you're A blood, of course, you can give it to another person with A blood. A person that's B blood, they can donate to someone that has B and somebody that has AB. But now let's think about the AB people. So if you're AB, who can you donate to? Well, you can't donate to O's because they're super finicky. They can't accept blood from anybody but O's. You can't donate to B because your A carbohydrates are going to mess with their bodies. And you can't donate to A because your B carbohydrates are going to mess with their bodies. So if you're AB blood, you can only donate to other people with AB. So in terms of like blood banks, if you are type AB blood, you are pretty much useless because you can only donate to one other type of blood group, which is great for you because it means you can get blood from anybody else. But if you want to donate blood, your blood's pretty useless on the whole. Finally, let's talk about the O people. Who can they donate to? Right, so if you have O blood, you don't have any carbohydrates on the surface of your blood cells, meaning that none of the other blood types, people with those other blood types, they're not going to react badly to you. So if you are O blood, you can donate to everybody. That's probably why you've heard of people referring to a person as a universal donor, right? So if you are O blood, you are the universal donor. You can give your blood to everybody, meaning that in blood banks, you are super useful because your blood can be put into any other human, which is really useful. But it also sucks for you because you can only get blood from other people with O blood. Okay, and then I want to we'll do a couple examples of this in a minute, but I want to just talk really briefly about the positive and negative thing. Right, so you probably heard, you're like type A positive or type A negative. And what that refers to is proteins on the surface of the blood. So the letter refers to carbohydrates, the positive or negative refers to proteins. So if you are positive, that means that you have the protein on the surface of your blood. If you're negative, it means you lack that protein on the surface of your blood. And those proteins work the same way as the carbohydrates. If you are positive, that means that your body's used to the protein. If you're negative, that means your body is not used to the protein, meaning that you could not accept blood from someone that is positive. So I want you to think about that and think about which blood type, like which letter in which positive or negative would be the universal acceptor, right? So who can get blood from literally anybody else in the population? So we already know that in terms of carbohydrates, it's AB blood, right? So they can get blood from anybody. And if you're AB positive, that means you also have the protein on the surface of your blood, meaning that your body is used to it all. It's used to both carbohydrates, it's used to the protein. It means that you can get blood from literally anybody else in the population and be fine. So now let's think about the universal donor. So we already know that O is the universal donor in terms of carbohydrates, and negative would be the universal donor in terms of proteins. Because if you're negative, it means that you don't have that protein on the surface of your blood cells. So with there being no protein there, other people aren't going to react badly to it. So O negative is the true universal donor because O negative blood doesn't have carbohydrates and doesn't have the protein. So anybody can accept O negative blood into their body in a transfusion and be fine. Okay, so that's a whole bunch about your blood type. And so if you know your blood type, think about that in the context of this chart in what I've taught you about. So let's do an example here to see what blood types the children might potentially have in this circumstance. So let's say we have a parent that has AB blood and a parent that has O blood, right? So let's think about what type of blood type could their children have, right? So the person that is AB blood, let's think about what letters they have, what their phenotype is going to be if you have AB blood, right? So you're going to have I capital A and I capital B, okay? So then the person that is O blood, they're going to have two copies of the recessive allele, two copies of lowercase I. Okay, so let's fill that into our handy Punnett square. By now, I'm sure you're all getting sick of Punnett squares, but this is really a truly useful way to kind of figure out how these things work. So we fill in the alleles on the top, and the alleles on the side, and then we're going to fill them in going across and going down. All right, so first we'll fill them going down. So I capital A, and then fill them in going down here. I capital B, and then we'll fill them in going across. So we have lowercase I there, lowercase I there, lowercase I there, lowercase I there. Okay, so we see that two out of the four boxes are I capital A, lowercase I. Okay, so that is going to be a child with A blood. The other two out of the four boxes are I capital A, I capital B, lowercase I. So that is a person or a child with B blood. So meaning in this circumstance, half of the children would be A blood and half of the children would be B blood on average, right? We would expect a 50%, for each child has a 50% chance of being A, a 50% chance of being B. Right, so let's think about that in terms of donations. Right, so let's say that this is really morbid, but let's say that these two parents, they're out for a drive and they get in a terrible car crash and they need blood. Can the kids donate blood to their parents? Well, in the case of the parent that has AB blood, that's great, right? Because they have a kid that would be either A or a kid that would be B. So the person that's AB blood, that's great. They can get blood from their kids. But the parent that has O blood, remember O blood is the universal donor. which makes them it makes it very difficult for them to accept blood. They can only accept blood from people with O types. So meaning they cannot get blood from their kids. So this poor old parent with O blood, they're dead because they couldn't get blood from their ungrateful kids. Whereas the parent with AB blood, good for them, they survive because their kids can give them blood. Okay, let's do another example. Let's say we have a parent that has the phenotype I capital A lowercase I, and the other parent has type O blood. All right, let's figure out what blood types their children might have. All right, so we have one parent that's I capital A lowercase I, and we have a parent that is O blood, meaning that they're lowercase I lowercase I. All right, so we're going to fill in another Punnett square because that's just what we do these days. All right, so we put the I capital A there, lowercase I there, and then we have two lowercase i's there. So as always, we're going to fill in going down. So I capital A, okay, and then fill in going down. So lowercase I, and then fill in going across. So going across there and going across there. Okay, so we see 2 out of the 4 boxes result in I capital A lowercase I. So that is a kid with A blood. Okay? And then 2 out of the 4 boxes are are lowercase I. So that is type O blood. Right, so each child has a 50% chance of being A and a 50% chance of being O. So let's use that morbid example again. So let's say that these parents crashed into those other parents from the previous example. And these 2 parents need some blood. And so they go to the kids and they say, kids, we need some blood. You know, we raised you. The least you can do is give us some of your stupid blood. So if you're the parent that is the I capital A lowercase I phenotype, that's great for you because no matter which child you choose, you can take their blood because all of their kids are going to be either capital A or A blood or O blood, and they can accept them both. For the parent with O blood, it's a little less certain. If it has happened that if they're lucky enough that their kid is type O, which there's a 50% chance of a child being O, then that's great for them. Then they can get blood. But if their kid or if their multiple kids all happen to be type A, well, that sucks for them because they can't get blood from the type A. So maybe the O parent lives, maybe they die. It depends on what the blood type is of their kids. But the parent that's I capital A lowercase I, so A blood type, great for them, they survive. Okay, let's talk about something a little less morbid than car accidents. Another way that genetics can be different from what we learned about from Mendel, from Mendel's four laws, is something known as pleiotropy. So pleiotropy is when there is one gene that is influencing multiple characteristics. So up until now, we've been talking about just one gene and one particular characteristic, right? So like there's a flower color gene, and that only has an effect on the color of flowers in a pea plant. But pleiotropy is when one gene influences multiple characteristics. And a good example of this is sickle cell disease, right? Which we've talked about a lot up until now. So sickle cell disease comes about from something on the hemoglobin gene, which we talked about back in chapter 10. Right. So the principal thing that the hemoglobin gene does is it transports oxygen. Right. So that is the principal kind of characteristic that is influenced by hemoglobin. Right. So that's the main thing. But then on top of that, hemoglobin, so that one gene, also has effects on the shape of the red blood cells, right? Which as we know now, blood cells can have this round shape or can have this sickle shape, right? So that's another characteristic. But it also has an effect on resistance to malaria, which we talked a little bit about. Essentially, if a person has these sickle cells in their body, it makes them less likely to get malaria. So that's then kind of another characteristic that is affected by hemoglobin. And we talked about how resistance to malaria is a lot of why is a lot of why sickle cell disease came about in the first place. Because essentially, because malaria, it's way worse to have malaria than to have sickle cell disease. So essentially, by having those sickle cells, it's a good thing if you're in an area where there's a lot of malaria. Okay, so that's pleiotropy. So that's one gene influencing several characteristics. Uh, However, we can have the reverse of that. Uh, and the reverse of that is called polygenic inheritance, right? So the word polygenic should kind of tell you what it means. So poly means many, genic means gene. So we're talking about many genes. So two or more genes affecting a single characteristic is what polygenic inheritance is. And a great example of this is height, right? So we know that humans don't occur in just either short, like Muggsy Bogues, or tall, like Manute Bol. There's a whole range of heights in between there. And so why there's that large range of heights has to do with the fact that there's many genes that controls just how tall you are. If it wasn't the case, then everybody would be either tall or short, but it's not the case. There's a variation in height, and that's because there's many genes that control our height. Okay, let's talk a little bit about the role of the environment here, uh, because we've been talking about genes up through now and how genes affect how we look. But in fact, the environment that is around us, right? So like the climate that's around us and the food that we eat and whether or not we exercise and what our parents, do and what our family is like. All of those things also have an effect on us, not just on our behavior, but also on our shape. So consider, for instance, uh, identical twins, right? So think about a set of identical twins that you know, right? Most of us know at least one set of identical twins, or at least maybe knew a set of identical twins when we were growing up in school. For most identical twins, people that know them can tell them apart. Uh, so even though those two twins have the same exact DNA inside of them, they have the same exact genes, there is some slight variations in their appearance and probably some major variation in their behavior. So even though they have the same exact genes, the environment has an effect that causes two people that with the same DNA to be different from each other. So the environment's very important. And so many characteristics, uh, they result from a combination of heredity, meaning the genes that we get from our parents, and from the environment as well. So a good example of this is kind of the nature versus nurture debate, right? So this is maybe something that more like parents tend to think of. Can you truly, alter the behavior of a kid, or is it just in their nature just to behave how they're going to behave? Well, in fact, it's kind of both, right? So there are innate things inside of us in our genes that that control us, but how we are raised also has a big effect on us. So a good example of this is height and weight. So these are things that are of course affected by genetics, right? So people that are tall tend to have kids that are pretty tall, and people that are thin tend to have kids that are thin. But we know that diet and exercise also have an effect on these things, particularly weight. Right. So if a person has a high fat, high carbohydrate diet, then they're probably going to have a greater weight than somebody that has a low carb, low fat diet. And a person that exercises is obviously going to have a very different shape from a person that doesn't exercise, right? So even though things like weight and height are affected by our genes, we can also change them a little bit with the things that we do, the things the things in our environment that we interact with, mainly our diet and whether or not we're exercising. Okay, so another variation of Mendel's four hypotheses that I want to talk about is what's known as sex-linked genes. So sex-linked genes, you can probably tell that they have something to do with gender. So gender then has an effect on whether a person has this certain sex-linked trait or not. And so this happens when genes are located on sex chromosomes, right? So we learned back in chapter 10, and I guess a little bit in chapter 8, that sex chromosomes are the X's and the Y's. And those are what makes a person genetically a male or genetically a female, right? So a person has an X chromosome and a Y chromosome, they are genetically male. If a person has two X chromosomes, then they are genetically female, right? So I'm using the adjective genetically, right? Genetically male or genetically female, because gender is a lot more than just what genes you have in what in what chromosomes you have. It is in fact possible for a person to have two X chromosomes but have all the characteristics of a male. And it's possible for a person to have an X chromosome and a Y chromosome and have all the similarities to a female. So there's a lot more to gender and how we identify than just what chromosomes happen to be inside of you. So I'll be kind of using that term genetically male or genetically female as we talk about sex-linked genes. And so these sex-linked genes are things that are on a sex chromosome. So it's a gene that happens to be located on an X chromosome or a Y chromosome. And the result of that is that which genetic gender you have will have an effect on whether you have a certain trait or not. So these are things that are usually on the X chromosome rather than the Y chromosome. The reason for that mainly is just that the Y chromosome is quite small. The Y chromosome is actually the smallest of all of the chromosomes in the human genome. It turns out it just takes a lot less information to make a genetic male having a Y chromosome than to make a genetic female having an X chromosome. Right, so all you guys, you're all very simple. So sorry to disappoint you if you all think you're really complex genetically, but us guys, we are just genetically a lot less complex than females are. So a good example of a sex-linked gene is red-green color blindness, and so that is the inability to tell apart red from green. So for instance, if you look at this picture here. You should be able to see a number in that picture if you can tell apart red from green, right? So can you all see the number that is in there? Sorry, I pranked you. That was actually what it looks like if you have color blindness, right? So if you're not colorblind, you should now be able to see the colors. I'm sorry, the number 7 in there, right? So you can see the green 7 amongst that. If you want to see what the world looks like as a red-green colorblind person, what you can actually do is you can just take a picture and upload it to any number of websites where it will then make it have the appearance that it would have if you were red-green colorblind. So you can see what the world looks like to a person that is red-green colorblind. So let's talk about sex-linked traits and colorblindness and talk about carriers. Right, so if a trait is on the X chromosome, I want you to think about this. Can a genetic male be a carrier for something that is on the X chromosome? Right, so think about it. Genetic males, what chromosomes do they have? Do they have two X's? Do they have two Y's? Do they have an X and a Y? So genetic males in general have a X and a Y chromosome, meaning that they cannot be carriers because they only have one X chromosome. So for something like color blindness, a genetic male is either colorblind or they're not colorblind. There's nothing in between because they only have an X chromosome. They only have one X chromosome, I should say. Whereas a genetic female can be a carrier because a genetic female has two X chromosomes, so they could have one colorblind allele and they could have one color seeing allele, right? So a genetic female can be a carrier, whereas a genetic male either is colorblind or isn't colorblind. So the answer to this question, as I just explained, is no, because males only have one X chromosome. Okay, let's do we're going to do two examples of this, so you can work through how to do problems including sex-linked traits. And by the way, when you see when you see questions either on a quiz or on an exam, I will tell you if I'm talking about a sex-linked gene. Okay, so for sex-linked genes, we're going to be using X's and Y's because it's related to X and Y chromosomes. But unless I tell you that it's a sex-linked gene, don't worry about X's and Y's. Get a lot of students that get confused by that, where they'll use X's and Y's for like blood types or for like cystic fibrosis or sickle cell anemia. Unless I say sex-linked in a question, don't use X's and Y's. So we have a colorblind male and a carrier female have kids. So let's figure out if the kids have any chance of being colorblind. First of all, I want to make sure that we know what the alleles are that are possible. So first of all, one possible allele is, we're just going to write that in as Y. Okay, so that is going to represent the Y chromosome. Okay, let me just erase that. So that's one allele. We can have an X chromosome that has the dominant allele on it. So capital B because we're talking about blindness, so I'll use the letter B. So that's another allele. And then a third allele would be X with the lowercase b attached to it, right? So then that has the recessive allele attached to it. So it's very important that you don't put any letters associated with the B, because color blindness is something that is on the X chromosome, not on the Y chromosome. So we put a capital B or lowercase b on X's, but not on Y's. Okay, so we're talking about a colorblind male and a carrier female. So let's put the colorblind male over here. Okay. Okay, and let's think about what alleles he would have, right? So we're saying that he's a male, so we know that he is XY. And we're saying that he is blind, meaning that on his X chromosome, we're saying that he's colorblind, so on his X chromosome, he should have a lowercase b. Okay, and then the female, it says that she is a carrier. And so first of all, we know she's a female, so we've got two X chromosomes. And if she's a carrier, that means the same thing if she was, say, a carrier for cystic fibrosis. It means that she's heterozygous. So she's got one capital B and one lowercase b. So it's really important that you know that if she's a carrier, which means that she carries that recessive allele, but she herself is not colorblind. Because she also has the dominant allele along with that recessive allele. Okay, so let's draw in a Punnett square. All right, so we have the female on the top, X capital B, X lowercase b. Male on the side, X lowercase b, and capital Y. So let's fill in the boxes going down. So first X capital B there, then X lowercase b, X lowercase b. And then we do the same going across, x lowercase b, x lowercase b. I'm sorry, I totally screwed this up. Here, let me go back in and erase. Wait, no, I'm sorry, I didn't screw up. Never mind. So then we fill in going across with the y. So we have capital Y there and capital Y there. Okay, so let's take a look at these kids. Right, so the first kid is x capital B, x lowercase b. Right, so first of all, is that a male or a female? Right, so that is a genetic female. And is that female color seeing or colorblind? So in this case, she's heterozygous, meaning that she can see colors. So she's color seeing. Okay. Then the next box we see is x lowercase b, x lowercase b. So it's two x's, so again, that's a female. And she has two copies of the recessive allele, meaning that she is color blind. Okay. And then with the next box, we have x capital B and y. So that is a male because there's a Y chromosome, or at least it's a genetic male, I should say. And because he has the capital B allele, that means that he is color seeing. And then finally we have the last situation, x lowercase b, capital Y. So this again is another male. In this male, because he has the lowercase b, he is colorblind. So in this circumstance, these two parents, right, they could have a colorblind daughter, a color-seeing daughter, a colorblind son, a color-seeing son. They essentially have all of the potential options with their kids. Okay, let's do another example. Okay, let's say that we have a color-seeing male has kids with a colorblind female. So again, we'll put the male on the side here, and he's color seeing, so he's XY, and on his X will be a capital B because he can see, colors, so he has the dominant allele. And then we'll put the female on the top, and it says that she is colorblind, meaning that she's going to be X lowercase b, X lowercase b. Because remember, for a female to be colorblind, she needs to have two copies of the recessive allele. Whereas for a male to be colorblind, he only needs one copy of the recessive allele. So we're going to fill in another Punnett square. All right. So we fill in the females' alleles there, the males' alleles there. All right, we're going to fill in going down. So we have x lowercase b, x lowercase b. Fill in going down, x lowercase b, x lowercase b. And then we fill in going across. So capital X, capital B, capital X, capital B. And then y there, and then y there. Okay, so we see that 2 out of the 4 boxes are x lowercase b, x capital B. So first of all, is this a male or female? Right. So the answer is female because it's two x's. And is this female colorblind or color seeing? So this female is color seeing because she, even though she does have one copy of the recessive allele, she also has a copy of the dominant allele. So she is color seeing. But she is a carrier, meaning that she could potentially have kids that are colorblind. So then the other 2 out of the 4 boxes are x lowercase b and capital Y. So that is going to be a male. And those males have the lowercase b with them, meaning that they are color blind. Okay, so what we see here is every child that these two have There's a 50% chance that the child is a female that can see color, and a 50% chance that the child is a son that cannot see color, right? So all of the daughters that these two have would be able to see color, and all the sons that these two have are not able to see color. So what this tells us is any female that is colorblind, if she has sons, all of those sons are going to be colorblind. There's no way for them not to be colorblind. So if that happens to be you, so if you're a female that is colorblind and you're going to have kids, if they're sons, sorry, there's just no chance that they're not going to be colorblind, that they're destined to be colorblind. And this brings up a really important point. With males, they only need one copy of the recessive allele to be colorblind, right? So these males, they just have that one copy right there in their colorblind. Whereas females have to have two copies of the recessive allele to be colorblind. So what I'm getting at here is that it's way more likely for males to be colorblind than for females to be colorblind. And so I want you to, think about the people that you know. Think about how many males do you know that are colorblind, right? And now think about how many females that you know that are colorblind, right? So pretty much all of you are going to be able to at least think of a couple guys that are colorblind, but probably not be able to think of any females that are colorblind, or maybe just think of a few. I've only had, I think, two students in all the years that I've taught that were both female and colorblind, right? It's quite rare for a female to be colorblind. Okay, so that is all I want to tell you about genetics, about what makes us who we are, about Mendel, and about inheritance. So in the next chapter, we're going to kind of switch to something new. We're going to get into the evolution part of the course. So until then, have a good day, and we'll see you next time in the next video lecture.